you. Send us your views, comments, and videos with your name and town to the One Africa TV's WhatsApp on 081 200 6659 or send an SMS to 555. One Africa TV. It just gets better.
I want us today to make sure that we have a real, clear understanding of different genetic problems. So I try to mix them a bit to see what do you have to read into a question. Okay? What do you have to read into a question? So when we look at the first question, it is always important to check what information is provided. The stages in sexual reproduction in a mammal. And from organ P, and a cell from the testis. They will go through a process, and then together they form a fertilized egg and a process, and now there's an embryo, and process Z will form lots of different types of cells. What is the name of organ P? Organ P, ovaries, because it was an egg cell or an ovum. Give the names of parts A, B, and C in cell 4. Nucleus, cell surface membrane, cytoplasm. What is the name of cell 3? It is a sperm cell. What type of cell division takes place in process Y and Z? Mitosis, because it is one cell that increase in number due to cell division. What was the name of process X, if you care to go back? X was to form a gamete, so that was meiosis, right? Now it was mitosis. Which two of the cells labeled 1 to 6 are genetically identical? And that will be after the ovum and sperm fused, fertilization took place and a zygote formed. That zygote then start to divide into an embryo. So the zygote and the embryo, 5 and 6, definitely was genetically identical. Good. The gametes, we know, was the sperm and the egg, so it was 3 and 4. The cell 6 change into a range of different cell types. The name for this different cell types was stem cells. And when they change into many different kinds of cells, it is due to differentiation. Because the structure of the cell changed for a special function, the cell differentiate to have a special function for specialization. Differentiation is what we can see happen to the cell. Specialization indicates the new functions that this cell can fulfill. Cystic fibrosis is a condition in which people suffers from the accumulation of thick and sticky mucus in their lungs. Cystic fibrosis. Now, here we see males and females that suffers from this, and I want us now to have a clear understanding of the female and another female. Those that are colored is unaffected. Those that's only a circle, it has cystic fibrosis. So here we see a female Trudy, so all the circles are female, and Sadia, they are unaffected, right? And Sarah and Angela and Kirsty are affected females. The male, when it's a square, we have the cystic fibrosis, males, and then when it is a filled square, they are unaffected. So we see that Liam is unaffected, Farad is unaffected, Josh is unaffected. Right. Gareth also unaffected. But Richard is affected. Good. So, use two pieces of evidence from the family tree explaining why cystic fibrosis appears to be a recessive gene. A recessive gene. So recessive means it is a gene that when the gene is paired with another recessive gene, we will see the disease, the phenotype of the person will be cystic fibrosis. When it has the dominant, not affected gene, that person will not be ill. You agree? So, when we look at Angela, we will see Angela has the disease. So Angela is FF, right? And Farad is big F, small f. Liam is unaffected. But Liam must have an F. Angela inherited a gene from her mother, which was a small f, but from her father also a small f, although he is not affected. So it is telling me that although that dad is not affected, he contains the gene. Now, and that is how I know if he can have a child with the disease, he contributed to the genes of Angela. And that then therefore he was not affected. So his small f, his recessive gene was masked off by his not affected gene. And therefore it must be recessive. Josh and Sadia is not affected. But they both inherited the gene. So they inherited the F gene. But they are not affected, so I know that it must be a recessive gene, because you can see they are not affected. Okay, right, so Trudy and Gareth want to have another child. What is the chance that this child will inherit cystic fibrosis? Trudy and Gareth, Trudy and Gareth is unaffected. But they have already 2 children that are affected. So, if we look at the phenotype of Trudy and Gareth, they are heterozygous not affected and crossing with heterozygous not affected. The phenotype of the parents looks like that, and when we look at the gametes, and please remember, we always circle gametes, we will see that there is the possibilities. And just to make it a bit easier to see the different fertilization, we see that we have offspring 1, 2, 3 that is not affected and 1 that is affected. So, we will have normal to cystic fibrosis, 3 to 1. So, is there a possibility? Yes, they always have a chance of 1 out of 4, or a quarter chance, or 25%, or a ratio of 1 to 3 for cystic fibrosis to normal. Take care that you indicate in which order you do give your ratio, please. Okay, the next then, who had the different combinations? Liam had a dominant and a recessive allele, Angela recessive, and Sadia also B dominant and recessive. Explain how it is possible that Farad and Angela could have a child with cystic fibrosis. Again, we said Farad is heterozygous. So Farad looks like this. Angela looks like this. If a kid inherit from Farad's sperm that F, from Angela, the only possibility will be to inherit a recessive F. Their children will have a possibility. And remember, when we have heterozygous, homozygous, then there will always be a 50-50 percentage, a 1-to-1% of having a cystic fibrosis or an unaffected child. Okay, let's look at the next question. This question is about the hereditary material of the human species. How many chromosomes are found in the human karyotype? There are 46. There are 46. How will the chromosome composition of a human be in a somatic cell? In a somatic cell of a female, it will be 44 plus XX. In the male, it will be 44 plus XY. In the gamete, it will look like this. 22 plus X is one of the male gamete, or 22 plus Y. So a male gamete has 2 options to divide. Just to conclude, the female gamete will look like this. 22 plus X. 22 plus X. Okay. Hemophilia is due to a sex-linked gene. Hemophilia is that disease that we talked about in the video of the king, the royal family, when you have a heat against your body, you can have internal bleeding, or it's very easily for you to bleed because your blood does not clot or coagulate very easily. So, the question is, hemophilia is due to a sex-linked gene. It is recessive and found on the chromosome. A woman who is on the X chromosome, it is on the X chromosome. A woman who is a carrier for hemophilia marries a normal man. What will be the possibility phenotypes of their children? Use the letter H and use a Punnett square with labels. So, if it is a recessive gene, the gene for hemophilia looks like this. You agree? So, if we are going to put in a Punnett square, let's just move down our Punnett square. I'm just going to clear here so that we can easily see the correct answers there. Okay, so we say we have a normal man and it is a sex. So the H is the gene for hemophilia, right? A normal man will have an X and a Y. Because it is only carried on the X, there will be a capital H. There will be nothing on the Y. That is the male. Okay, you agree? Male. So let's write down the male in there. And the female is a carrier. So she will have the normal and the recessive. Because we are talking about gametes, we need to put in our circles. Right? We need to state that this is the male and this is the female. Right? And we need to state that this is meiosis. That makes this possible, please. Okay. Good. Now we are going to look at fertilization. And here we will have X, H, X, H, X, H, and X, Y. We will have X, capital H, X, H, and X, H, and Y. Okay. What will the possibility phenotype of the, what will be the possible phenotypes of their children? So their children will have there, we have a homozygous normal child. And what will it be, male or female? It will be a female, no? It will be a female. Right. This one will be a heterozygous. And when you hear the word heterozygous, in the case of a recessive disease, we actually know that it is a carrier. A carrier for hemophilia, and it will be a female. Right? These two are covered then, there, and there. Now, this will be a male, and that male will be a normal or normal male, right? And this male will be a hemophilic male. Okay, I hope that you now with sex link we see not only who's got the disease and who does not. We also see male and female in this answer. Now, okay, right. A phenotypical normal man who has a hemophilic brother marries a normal woman who is not a carrier. What is the probability that any of their children will be hemophilic? Now, a man can only be either a sick hemophilic or a healthy person. You agree? So this man is normal. And his wife is normal. Yes. I had to use a H for hemophilic. Let's do this again. Going to try and get us that very thin line now. Okay. So a male looks like this. H for normal and a Y. The female is normal and she's not a carrier. She will see she's H and H. They are as normal as can be. So what is the probability? They will have 0% chance to have a child with that. Okay. Color blindness is an X-linked recessive trait. If a normal-sighted woman whose father was colorblind, what is the probability that they will have a son who is colorblind? Use the letter B. So, colorblind is a recessive trait. So we will have a small b, right? A normal-sighted woman will look like this. On the X, she will look like this. Because this X of this normal woman came from her father. So this woman is definitely a carrier, unfortunately, for her children. She's definitely a carrier, nah? Because her father was colorblind. One X she got from her mother, but the other X came from the father. The father had only one X. So it was then inherited, not given. That's wrong to say it was given to, because it's not giving. He makes his children inherit his genetic profile. Now, so now she married a colorblind man. So we have to start with the phenotype, colorblind father, crossing with a normal phenotypic, the genotype, small b, the gametes will look, and we have fertilization, and then we have the offspring genotype, and we have the offspring phenotype. Good. So the fertilization will look like this. X and a X, B and a B. Immediately, I am trying to tell myself that is a female. Right. Be careful. Then the next one, that and that is and X, B, X, B, and that is again a female. Then I have a Y, so that will be a XB and a Y, and there will be a XB and a Y, which will be the males. So what is the probability that they will have a son who is colorblind? They have a possibility of 2 sons, right? You agree? And now there's a 1 out of 1. There's a 50% that one of their sons will be color. Let's continue. In man, muscular dystrophy is a condition in which the muscles waste away during early life and may result in a shorter lifespan. It is due to a sex-linked recessive gene. A certain couple has 5 children, 3 boys and 2 girls. The boys are 1, 3, and 10, and the girls are 5 and 7. The oldest boy shows symptoms of the disease, but none of the parents shows any symptoms. If the boy is showing symptoms and it is a recessive, it means that it is a sex-linked, so it means if we use a small m for muscular dystrophy, the boy will look like that. And the boy can only inherit this X from whom? From his mother. Because remember, the mum is this. You agree? The boy do have the symptoms. So, he must have inherited this from his mum. What about the dad? The dad is X, Y. Right? But that, if this boy, the only possibility for him to got this disease was from his mother. Now what's the question? You are the family physician and they come to you for advice. What would you tell them about the chances of the other children developing this disease? So, now we will need to make a Punnett square or something in order to help us to sort this problem out. So our Punnett square, let's insert a Punnett square here. The mother, the father, this, because he does not have the gametes. Here, this girl will be homozygous normal. Right? This will be a boy, and this boy will be homozygous, it will be normal. Now, here we will have a girl again, 2x, but she will have a capital M, small m. Capital M, no disease. X, capital M, no disease. Small m, capital M, this will be a carrier girl. And the last one will be a boy again with a small m. This will be a boy with the disease. You see, so when they have a 1 out of 4 chance, 1 out of 3, 1, 2, 3 chance to have a ill child with muscular dystrophy. And it will be the boy. Therefore, class, they normally says that it is the mum that carries the disease to her son. If the mother is a carrier and the dad is healthy, it's only the X on the mum that will be the gamete that's carrying it to the boy. I hope that you do understand that. What is meant by sex-linked genes? And this was now on page 501. What is meant by sex-linked genes and sex-linked disorders? A sex-linked gene, a gene found only on sex chromosomes. So we saw in these questions hemophilia, color blindness, muscular dystrophy. These are all diseases that is a sex-linked disease because it is attached or on the X chromosome. Male do not have two X's but only one X. So if a male inherit the dominant gene, he will have the condition. If he inherited the recessive, on the Y there is nothing to mask it off. He will be healthy. If a female has one of the dominant and one of the recessive, she's a carrier. Okay, right. Any disorder that is determined by the sex chromosome or by a defective gene on a sex chromosome is a sex-linked disorder. Then one last question, help us think. A man with normal color sight marries a woman who is carrier for color blindness. What is the probability for their children in percentage to be color blind? So let's write in the male. Going to put the male here. So the male looks like this. And the female, the male X, N is the one gamete for the male, and Y is the other gamete for the male. The female X, N, X, small n. She is the carrier. So this child will be an X and an X, so it's a girl. Capital N, capital N. So normal vision. N, another girl. So these two here we will have girls, right? There we will have now an X, N, and a Y. X, small n, and a Y. And this will be the boys. Now, there's always a 50-50 percentage to have a boy or a girl, because we have a heterozygous and a homozygous crossing, always 50-50%, right, or 1-to-1. But to have colorblind children is 1 out of 4 is 25%. Our answer there will be 25%. The next question, which we are also going to use this, how many daughters will be normal? 1 out of 2. So 50% they care. 50% of the daughters. This was not about children. And if 50% is normal, in this case, we will have 50% carriers. What if a daughter marries a colorblind man? What if the daughter marries a colorblind man? And I just want to make sure he's a carrier. So the daughter who is a carrier marries a colorblind man. Now we have exactly the same scenario. We have the daughter who is a carrier. Please remember this is gametes and circle them. Marries a colorblind man. So small n and a y. Ne? They will have a carrier daughter and they will have a colorblind. They will have a possibility to have a colorblind daughter. And their sons, they may have a normal son, but they also may have a colorblind. So they have a 50% chance of having a colorblind child. No, they have a 50% chance of having a colorblind child because 2 out of the 4 children, that gives me 50%. Okay, right. And the last question is, color blindness is more common in men, as I have explained, because it is carried on the X. Man has only one X chromosome, and therefore there is not another one to mask off. Okay, grade 12, please make sure that you do understand this work through all the different possibilities of how inheritance problems can be asked.
One day we went to I think it's the opening of Oshakati Stadium. Then I was a lead singer there. Then this DJ Remind came to me and was like, "Hi, don't you want to be a solo artist?" I said, "What is solo?"
Ladies and gentlemen, my name is DJ Siambra Mazukiro Mkwampiri Perez Kampuka. Please return from the might of this region.
Happy Friday, what's up? It's Friday, happy Friday.
Abdallah is joining us in the hot seat.
She said it right. She said it right. She said it right.
Welcome to the tribe.
Thank you so much.
Hi to Holland, Namibia. Welcome to the tribe on One Africa TV. My name is Shay the Goddess, and I keep you company every Friday, along with some of Namibia's biggest artists right here on this platform. What do the letters DNA stands for?
deoxyribonucleic acid. Where is most DNA found? In the cell of a eukaryotic, and that is the nucleus. A prokaryotic is in the cytoplasm. And the red blood cell has no DNA. State the names of the bases. Please spell correct. Or so that when I say your name, that it is correct. But higher level, they are strict on specific scientific words that you have to spell it correct. Okay, so if one must be thymine, because thymine links with adenine, and guanine will link to cytosine. The component X, I want to see deoxyribose, because you are higher level. It's not only a sugar, it has a specific name. And then, of course, Y, the phosphate group. Thank you. Many of you remember that. Purine is guanine and adenine. And I realize that a way to remember, the pyrimidines are the smaller ones, the ones that is only one ring. Yes, somebody said the purines are 2 rings. Nah, the pyrimidines is only 1 ring, but we can also remember thymine has a Y and cytosine has a Y. And that will then help us to remember pyrimidine, thymine, cytosine. Now, that is also a way to remember those 2 different ones. Then, describe why, the first question there was, diagram shows a process taking place and will change the DNA diagram. The process visible is replication. There strand 1. Why is it important for this process to take place? We need identical copies of the genetic instructions to be passed on to a daughter cell. So during cell division, if you say during cell division, identical copies must be made for the daughter cells. That is also a good answer. Then here, for 6 marks, I think that it is important to say the DNA will unwind and unzip. This happens by helicase, and I thought that earns a mark. Break hydrogen bonds between the nitrogenous pairs. That you cannot just say the hydrogen bonds break, because unfortunately there are hydrogen bonds between other parts, inside the cytosine, to make that cytosine circle. They can be hydrogen bonds. So the hydrogen bonds break between the complementary nitrogenous pairs or between the nitrogenous pairs. I need to know where the hydrogen bond is that will break. Allow the strands to separate. Bases are exposed. There we can see the bases are now exposed for a nitrogenous base to attach. Now, bases are exposed for the free nucleotides with its nitrogenous base to attach. Complementary base pairs happens at A to C and C to G, and ligase help to form the backbone. DNA polymerase can be in the next line. DNA polymerase links the nucleotides to identical DNA molecules will form. So make a simple drawing, and what I think was important for that drawing is that you have now strand 1. Nee. So here we have strand 1. There was strand 1. So you draw there and you show me the nitrogenous bases. But that is not enough. I need to see how are they going to attach. So A will attach, and this is DNA replication. So this is another DNA strand. Nee. And A will link to T. Yes? So that will be a blue one. There's another A, T. And T will link to A. That one will be a red one. And then G and C. So let's just make C. Let's make C green. C. That one looks like, and this. Okay. So it will link to a yellow, and G will be green. You agree? But you cannot use color, so let's rather write in there A link to T, T to A, C to G, G, T to A, G is yellow, so that's a C, C, G, and T. That is how the other side will look. Make a simple drawing to show the end of this process for strand 1. So there I just link them. So the end of the process will look like this. 8, and I will have A, T, T, A, C, G, C, G, T, A, G, C, C, G, A, and T. And this is the end of the process. So for your 2 marks, I would have checked, do we have them all linked? Do I have this ladder diagram, and are all the bases correctly linked? Describe why this is called a why this process is called semi-conservative. Then the answer there will be the words that one original strand serve as a template for the new strand, and the new strand, that will be my second mark in yellow, new strand to keep the original, to use as a template for the original code. What does it mean that the 2 strands are complementary? A will link to T, G will link to C with a hydrogen bond. Then we have the diagram. So if this is the DNA, the mRNA will be linked with a complement, right? Between those 2, it will link with a complement. That means the T of the DNA cannot link with a T. That is not complementary. They cannot link. They do not fit into one another. So the T will become U in the RNA. Yes, in DNA. In DNA, T links to A. T links to A. A links to T if we talk DNA. But if we talk RNA, we know there are no T's in RNA. The T is substituted with a U. Aha. So our answer is A-U-A. Right? A-U-T-A. The codon then on the tRNA will then be U-A-U. Okay, just check. So mRNA is the complement of the DNA. The complement of DNA in RNA language will not have a T in it. Indicate the location in the cell where transcription is in the nucleus. Transcription is in the nucleus. Translation in the cytoplasm, or if you say ribosomes, and mRNA is produced, but in the nucleus. Good. Use the diagram above and explain the terms transcription, the process of constructing a messenger RNA molecule using a DNA molecule as a template with resulting transfer of genetic information to the messenger RNA. Translation, the process of forming a protein molecule at a ribosomal site of protein synthesis from information containing in the messenger RNA. So I want to see the word protein molecule, a ribosome, and messenger to give you all your marks. Now compare the differences. And there on my slide you will see the differences. DNA is a double strand while RNA is a single strand. But I ask in a table, nee. So you have to show me your answer in a table, please. If the question was not in a table, you see you cannot say DNA is double. And then I have to think what RNA is then? Triple? What? RNA is a single strand. Nee. So that I want in a table, please. DNA has T, thymine, while RNA has U. DNA has deoxyribose sugar, while RNA has ribose sugar. RNA is also found in the cytoplasm as well as in the nucleus. Well, DNA is only in the nucleus. That's 4 different ones. You have more. What are the similarities? Okay, let's study. Both are nucleic acids. They are made out of nucleotides. Both have a sugar phosphate backbone, and are found in the nucleus. So there are also 4 similarities that you can quickly remember. So, thank you, great
On the Gold Coast of West Africa lies the peaceful and diverse nation of Ghana. I'm pretty close right now to the geographical center of the Earth. It is also one of the last pockets for elephants in the wilds of West Africa.
You know, every country in the world just about has cattle, sheep, goats, horses, donkeys. But it's only Africa that has the real megafauna, the charismatic stuff which people in the rest of the world want to see.
The passion that the staff have for the job.
There's value to a bird which brings birders to a place like this. I mean, this is a birding paradise. We see an intrinsic appreciation and joy from seeing the wildlife out here, but not everybody does.
You can't get to Moli and then will not be passionate about what we do, because you'll be the only one left. Everybody is passionate.
The only worthwhile war, the war against destruction, the war against loss, the war for peace, for balance, for inclusion.
It's out of my way. Buffaloes, elephants, tigers, attabis are still in existence.
If Moli fails, then Ghana is without wildlife.
Join me as we journey to the untamed wilderness of northern Ghana to meet these last defenders.
The people within the law enforcement agencies don't really know you're talking about a roan antelope. Somebody thinks it's just like a goat.
We always love to hear from you. Send us your views, comments, and videos with your name and town to the One Africa TV's WhatsApp on 081 200 6659 or send an SMS to 555. One Africa TV, it just gets better.